This is about the specificity of molecular recognition. It is based on experiments that concern DNA oligonucleotides. Now, in the biological cell, molecules bind to their counterparts very specifically. Usually, binding is described by an affinity or binding constant.
The orange molecule binds to a receptor with binding constant K1, the green one with binding constant K2. Here we ask what happens if now both compete for the same receptor.
Naively, one would expect that the occupancy in competition is just a function of the constants K1 and K2. Say if K1 is 10 times higher than K2, then there is a 10 times increased probability for 1 rather than 2 to bind. Here we show that in our study this is not so. In fact, the weaker binding molecule is hardly detected. Its binding constant is diminished by a factor of 1000 in the presence of the competitor.
Here we study DNA oligonucleotides. DNA is made from a succession of 4 bases, A, T, C, G. A single-stranded DNA binds to its complementary counterpart to form the famous double helix. If a perfectly matching strand competes with a mismatched strand, we see that either the individual binding constant Ka and Kb describe the outcome, or the mismatched strand exhibits 1000 times weaker affinity, while the perfectly matching one binds as if the other one wasn't present.
Now, how can that be? Well, due to thermal excitation, there are many binding configurations, and they all contribute to the binding constant. If the mismatch is marked by the arrow, only the bottom configuration is accessible, the other 3 are not. This means the perfectly matching strand has a certain probability to bind into a mismatched target probe pair, since the mismatched target does not occupy the probe in full in terms of microstates.
In thermal equilibrium, if you write the complete energy of the system, it looks like this. The total Gibbs free energy of the system is given by single occupation of the probe strand and the term that takes into account the interaction of A and B, plus an entropic contribution.
Consider the total Gibbs free energy as a function of probe occupancy. Normally, the minimum is given by the ratio of the binding constants. If the statistical weight of the interaction term becomes overcritical, however, the graph transforms to opposite curvature, meaning that full occupation by the better matching target corresponds to the minimum. Well, the complete theory is a little bit more involved, but in a nutshell, that is the main idea. Surprisingly, this may apply to a single mismatch in competition with a perfect match already.